A warm welcome all back. Thank you for coming. There's 2 more sessions before a well-deserved apéro. And I would like to first of all thank Daniel for organizing this session, and I'm very pleased to share it because it's a topic that is very dear to my heart, design of clinical trials. And I'm very pleased that we cover really a lot of topics in clinical trial designs. We start with randomization, which is at the core of most trials, and then we go to more confirmatory trials, group sequential, adaptive, fixed sample size trials, and then finally we go back to early phase trials. And our first speaker is Diane from Roche. Please, over to you.
Thanks, Marcel. Thank you, everyone, for coming back after the coffee break. My name is Diane, and I work at Roche as a data scientist. This work was done back in the day when I was still working in Aachen. This is my funding statement. And when I joined biostatistics, coming from mathematics, the first question that was posed to me was, could randomization, the actual randomization scheme that we use, have an impact on the results of a randomized clinical trial? And of course, you would think it shouldn't. It shouldn't. Like, if that had an impact, something's wrong. And what's happening is that in the literature, there's a lot about different assumptions that can have an impact in the way that the randomization procedure does make a difference in the results of our clinical trials. And so the problem was that there was no uniform way to study this issue. And we looked at all the different publications, but they all did things in a slightly different way. And so there was not a good way to study this problem. And so we created a framework. In the framework, we mainly wanted, maybe I should stand more in the middle. Mainly, we wanted to have a comprehensive list of available randomization procedures. We wanted a variety of assessment criteria, kind of all the different ways that can impact the results of a clinical trial. We wanted to integrate the design and the analysis in a nice, uniform way. And we wanted to also have it fully unit tested. We wanted to have it well documented, and we wanted people to actually use it. So we also needed to make it fast. This whole framework is implemented in the RPACT randomizeR. Thanks to the few people who just came to me in the coffee break to have a little demo. You can all look at this package on CRAN. It has 14 available randomization procedures, including the well-known permuted block design. Also, the complete randomization is pretty well-known, and random allocation rule maybe. But then the others on the list are not as well-known anymore, although they have been around for a couple of decades. They're also all described in this book that I'm citing here. You might think, why do we need this? How are they different from permuted blocks? And so I'm bringing you a small visualization here in this picture. You can kind of see all the possible randomization sequences that can occur if you just flip a coin for 8 patients. So if you flip and get heads, you take 1 step up. That might be the allocation to the experimental group. If you get tails, take 1 step down, and you get an allocation to the control group. So here in blue is one of the possible randomization sequences. And of course, what you can see is if you get tails every time, you end up with all the patients in the control group, and that's what you don't want, right? Would be a really bad clinical trial. And so, so what's usually used to avoid that is the permuted block design. And here in this example, for 8 patients, we have blocks of size 2, 2 patients in each block. So you take 1 step up to the experimental group, and then the next one has to go to the control group, so that you always have balance after 2 patients. Problem with this is it's super predictable. And so there exist a variety of different ways to make this less predictable. You can just say, for example, in this procedure, let's not exceed an imbalance of 2 patients. So after 2 in the same group, you have to have at least 1 to the other group. And the thing that you see here is that it doesn't go through the origin, so through the origin each time, and that means that you have less restrictions overall and less predictability. We implemented this in the way that each of the randomization procedures has this object called PAR. So here is for a big stick design PAR, so parameters, where you can then pass all the parameters to it, and it's basically an S4 object that is a container for the procedure's parameters. And then you can use this genseq function on this object to create the randomization sequences, and the getRandList function is just basically an assessor function to print the sequence in a nice way. There are plenty of more procedures. They're all described in the paper. Yeah, so next step was we want to have a multitude of assessment criteria. Like there are plenty of ones in the literature. They're always a trade-off between randomness and balance. One important one is selection bias. So that is often modeled using correct guesses. And the way that works is if you were to guess at each step which treatment is the patient allocated to, the first one here, for example, you would not know because it's basically a coin flip, but then once one is allocated to the experimental group, you will know that in the end it will be balanced again. So you can always guess the group that has occurred less frequently, and so in the end you will always guess one more time correctly than incorrectly in this permuted block design. And now we want to integrate the design and the analysis. So we have an assess function and a compare function. In the assess function, you can add all the different assessment criteria that you're interested in and assess one randomization procedure towards all these different criteria. And in compare, you can compare various different procedures towards the same criterion. So for assess, I have one example here with the correct guesses. With that convergence guessing strategy that I just showed you on the earlier slide. And then if you use that assess function, you get for each randomization sequence in your specific design that you selected, on the one hand, the probability of that sequence for the given design, and also the proportion of correct guesses for that sequence. And then you can get the weighted average of all those sequences. And in that way, you will know how on average, what's the proportion of correct guesses on average for your design. For the compare function, you basically now can select the whole list of randomization procedures that you want to compare. Let's say random allocation rule, permuted block design, and big stick design, and the metric towards which you want to compare them, and just add them to the compare function. And it also has a plot method where you can get a nice plot for your randomization methods. And what we see here in this one is that the big stick design is much less susceptible to this correct guesses compared to the permuted block randomization and the random allocation rule, which is exactly what we would have expected when we looked at the random walk earlier, because we saw that it has less returns to the origin. So on what we see is that the distribution is also more favorable, so it has less, or it has a lower proportion of correct guesses. In summary, I think I did not change the world by creating this randomizer package. I wanted to make an impact when I joined Biostats, but at least the package is now available on CRAN and it's tested and it's also fast. There's still a long way to go though. As you all know, those other methods are not widely adopted in the industry. They're also not at Roche, so to my knowledge, the package is not used at Roche yet. And with that, I thank you for your attention. If you have any questions, feel free to reach me at the apéro or email me. Thank you.
Thank you very much, Diane, for this very nice and concise talk on randomization, perfectly on time. The next presentation is by Gernot Wassmer on the R package for adaptive clinical trials. Over to you, Gernot.
Thank you very much. Actually, I apologize for this very short title in the announcement. You will apologize for your long title. Actually, it's RPACT, Confirmatory Adaptive Clinical Trial Design and Analysis. And to start with, with this very short introduction, group sequential designs are designs where interim stages are performed, and these were introduced in the late 70s. They are quite popular at the moment, and software exists starting from SAS, EAST, Atlan also, nQuery, PASS, and there are also R packages, for example, GS design, and also, as you will see, our package RPACT. Confirmatory adaptive designs additionally allow a data-driven redesign of the trial under control of the type 1 error rate. These designs were introduced in these very seminal papers by Peter Bauer and Bauer and Köhne in 1989 and 94 of the last century. And the main idea is not to pool the data, but to combine the p-values from the separate stages through a predefined combination test. And that's more or less the clue how these designs differ from the conventional group sequential tests. And these designs are already conducted and are also more or less recommended or at least considered in regulatory considerations. And I would summarize as follows: if conducted carefully, then these adaptive designs will be accepted and can be used in practice. So, what's RPACT? We distinguish between RPACT written in small letters and RPACT written in capitals. The small letters RPACT is the package. It's a comprehensive validated R package for the design, simulation, and analysis of confirmatory adaptive group sequential designs, those which are more or less those that are summarized in this book written by myself and Werner Brannert, published by Springer in 2016. And all the contents can be seen in this web page here. The company, however, is the location where we try to earn some money. It was founded in May 2007 by myself and my colleague Friedrich Palke. The idea is an open source software development with the help of crowdfunding. So we're looking for industry, more or less, or people who give us money, and we say we will develop such a package. We are very happy that we are currently supported by 22 companies where we conducted a service level agreement. And you see here the list, and the starting point was Boehringer Ingelheim. That was exactly who gave us the idea of doing such things, and the idea was actually coming from Hans-Jürgen Lomb, who's sitting here in the first row. But you see, the last our last customer is Pfizer, and we are happy to have him on board as well. One essential point is that we are giving annual trainings for free for these companies, and at the moment we gave, I know how many, 20, we gave 78 presentations up to now. So we also have a cloud, or that's more or less a Shiny app, but we consider it a bit more generally. It's a graphical user interface, of course, and this Shiny app provides an easy entry to RPACT, and it's starting also for you to create R Markdown and Quarto reports. We support Quarto, and that might be interesting for you to write reports as well. It's very helpful to learn or demonstrate the usage of RPACT in a very user-friendly and intuitive way. As you see here from this screen here, or as an example, you see here the output of a very easy application, a group sequential design with 3 stages, and just here you see the output, the possible plots, and so on and so forth. That's typical Shiny app, but the cloud should be more general in order to also create such kind of company structures within this cloud. The package itself was released on CRAN in October 2018, I'm sorry, and up to now we have 25 releases. The last is 3.4.0, which was released last week. And we also have RPACT on GitHub. And you see here in the download stat that it was an increasing download over the years, and it's more or less comparable with the downloads of GS design, which is around 1,000 or 1,200 per month. RPACT also comes with a lot of vignettes. These are examples for the application, and you see here the first is a step-by-step tutorial, and the second is a very introductory design, and I selected these two because the first is a very introductory vignette to show well how to start with. And Daniel Lakens, who more or less wrote this vignette, is a psychologist, and he said RPACT is great also for use in such another area as in medical statistics. And the second example is because I wanted to show that Marcel Wolbers from Roche also helped us to write these vignettes, and I think this is a very nice vignette to introduce the RPACT. And you see also that with Quarto we could very easy or very clearly structure the content of these vignettes. So what's the functional range of RPACT? It's for the design, the sample size and power calculation, the simulation and the analysis for typically conducted clinical trials that are designed for continuous and binary endpoint. And you see here typical group sequential designs, Wang and Ciatis, O'Brien and Fleming, Pocock designs, and so on. And although these more or less now conducted p-value combination designs, inverse normal design, and Fisher combination design. But also a design with no interim stages can be formed. That's simply a fixed sample design then. And for that, RPACT is also, I want to emphasize this, RPACT is also a program where fixed sample designs can be planned. And planned for testing means, testing range, and survival designs. And in that case, we have very I would say specialized situations included with peaceful accrual time and intensity, a flexible follow-up time specification, and so on and so forth. That's quite general. The simulation tool is more or less for assessing these chosen designs and also the adaptive enhancement. So to say, the sample size reassessment, how does the sample size reassessment change the power or the expected sample size of these designs? It's also applicable for treatment arm and population selection designs, and this is not only for the simulation, but also for the analysis for a concrete data situation. Let me, at the end, briefly say something about the package concept. It's only one package, but we wanted not to be here like on this picture, it's not clear what is done. It should be very clear. The package concept is performed as for a typical workflow, how a clinical trial is conducted. First, we start with the design, and then we may try to compare different designs, and we calculate the sample size for a specific endpoint at hand, and we might simulate specific characteristics through simulation or through calculation, and then at the end analyze the design. We define a design and do something with the design. And this is done throughout the design. As I have seen, in the randomizeR, you also use the lower camel case, right, throughout the package, and that's also what we do. That's our concept. We want to focus on the usability. The functions and the arguments and the object should be as self-explanatory due to the names. So get design, group sequential defines a group sequential design, get design characteristics, calculates the characteristics of such a design, and so on and so forth. We also have, typically we have utility functions. These are, for survival designs, the followings. We also have this function getObjectRcode. That's from an object, from our object, from a created R object, we can retranslate the corresponding R code. Test package is used for the installation qualification on a client computer or a company server, and that's more or less my last slide here. Stands in connection with our concept of validation. We have a formal validation approach which was inspired by GAMP5, so it's good clinical practice, not clinical, but for what stands AM, I actually don't know. GP, it's good practice. And our test package, our test package has around 30,000 unit tests at the moment. We are using test package with the teststat functions, and the unit tests refer to the functional specification defined in our huge documentation, and in our huge validation documentation. And this validation documentation is only accessible for our customers. That's the difference between the normal user, free use, and this is only for our customer. That's it. Thank you very much.
Thank you very much, Gernot, for a nice presentation on a package that, as you have seen, is quite dear to me as well. And our final speaker before we go to the panel is Lukas Widmer, and he will talk about how to go fast, actually how to go really fast in the context of patient phase 1 designs. Over to Lukas.
Thanks a lot. Is this? Yeah, it's working. Very good. Let me just turn on the pointer as well. Yay, excellent. So yeah, this is a very long title, but I'm trying to show you a little bit about how we went faster with our patient adaptive design simulations in the context of phase 1 oncology. This is joint work with Sebastian at Novartis and Michael Meyer, who's sitting up there from POSIT. And yeah, quick disclaimer, what you'll hear is our opinion and not necessarily that of Novartis. So how many people here are familiar with phase 1 oncology trials? Show of hands. Okay, maybe a quarter. That's pretty good. So for the others, phase 1 oncology trials, they're safety trials, but they're a bit different in the sense that the volunteers that we have in those trials, they're patients whose cancers have progressed despite standard treatment. So this is third line plus, basically sort of last resort. But we still need to limit the risk of overdosing. So this is a first-in-human trial. So we haven't been in patients before. So that's something to consider, risk of serious side effects. And conversely, if the drug actually works, which we don't know at that point, we also want to avoid being subtherapeutic as much as possible. So yeah, in that case, if it works, and we're here, then lack of efficacy leads to death. So that's also something to avoid. So ideally, we want to get to this target range as quickly as possible. And that's also the goal of these trials, to systematically increase the dose as quickly and safely as possible to determine the safe dose range for further study. So how do we do that? These trials are basically model-based dose escalation with really small sample sizes, and we'll just get into how small. And it's a really busy design stage. So what we start with typically is prior, and this is no human data, preclinical data, really expert knowledge, trying to come up with a dose response curve for serious safety events. And then determine a safe set of predefined dose levels and sample a cohort of patients, 3 to 6, so it's very small, for this chosen dose level. We then observe so-called dose-limiting toxicity events for this cohort, so really severe ones, that would actually will cause you to de-escalate or stop the treatment for those for those patients, and then update this Bayesian model. So this loop basically goes on until we've come up with a safe dose range. So if we want to analyze these designs, we also have to simulate this loop, and this is updating a Bayesian hierarchical model that potentially has a lot of dimensions, so this is not a cheap step. And originally, this actually needed days of compute time. So yeah, my R code is slow. What can I do? This is the part of the presentation where I will hopefully present something that is more broadly applicable than phase 1 oncology. The quote from Hadley, yeah, it's a language. R is a language optimized for human performance, not computer performance. And we had heard a bit about speed this morning as well in the keynote. So we want to be able to iterate on our designs, and days is just too slow. So what do we do? And the way we went about this is basically by first gathering some data. So we measured, where is this code actually slow? And there's really a trap to avoid prematurely optimizing the part that you think is slow. It really sometimes isn't. And then optimize once we have data. So just to give a bit of context how this looked like in 2019 when I started, we had a multi-day runtime for around 10,000 simulation replications. And each of these simulations fits this Bayesian adaptive model for like 10, 20 times, unless it's super toxic and you stop early. So, okay, we had a high-performance compute cluster. Michael Meyer was at Novartis at the time. So we used batch tools to run this on the HPC, which made it 50 times faster. And we were like, okay, 1 to 2 hours on 300 to 600 cores. That's already better. But still, it's not, if you want to iterate on these designs, it's really not satisfactory. And still quite a lot of compute, so also a lot of CO2 produced and really a relatively high load on the HPC. So the package we use, this is also on CRAN. I'm not going to talk about this in too much detail. We optimized that and found out that, okay, we can drop the normalization and merge some data and be really more efficient by doing less work, but equivalent work. And this only bought us factor 2. And we're like, okay, it's now around slightly under an hour on 300 to 600 cores, but it should really have given us more. And we found out by measuring again that, okay, this compute time is now dominated by aggregating results on the head node, which only uses 1 core. So we use 600 cores. And then after like 10 minutes, we use 1 core that just reads files and writes files and computes metrics. And we're like, okay, this has to go. So, and that's also a little bit what I'll demonstrate now. We'll rewrote this with cluster MQ. And what this brings is that, yeah, we can, be much more efficient in terms of overhead. I'll just detail that a bit. And yeah, we could also construct much more of the results on the workers. And now we're another 30 times faster, 1 to 2 minutes on 200 cores, which is 3,000 times faster and 200 times more efficient. So it's like the point where you can go for a coffee and have results and iterate on your design. So that made us happy. So how do we do that? I'll just give a very brief intro into how cluster MQ works. This hides a lot of the magic, but it's all you need to conceptually understand what it does. So you load the library, define a function you want to compute. I mean, this is very inexpensive to compute, but a short example. And you have 6 values of your design variable that you want to vary and some jobs that you want to run. And then this magic Q function does the following. So you have your main node where you start up this computation. There's a scheduler for the high-performance compute system that depends on your system, really. But cluster MQ takes care of that, in principle. This then fires up these 3 jobs, and then each of these workers gets 2 of these function calls, because we have 6 X values and 3 jobs. So these get load balanced across, so not all of these workers will actually have something to do, and otherwise they'll close down. You'll get results, and things will shut down. And there's really a much lower overhead here. This is from the cluster MQ. It's on the vignette, I think, and also on the paper. And if you have, I don't know, sometimes 100,000, we already have, we have, what, half an hour of overhead versus basically a second or 2. This really makes a big difference. And it's really just, getting rid of results files on network shared storage, and also having persistent workers. So these R sessions that I showed you before, they're persistent and they just get new work. So much lower overhead. There's also some work by Willy Landau that I'd like to just plug here that I think is quite interesting, which is backends for targets based on a similar protocol than 0MQ. It's a successor in an even lower level language. So that's something to keep an eye on. And I also mentioned that I'm going to go a little bit into best practices. So this is regarding that. Each of these parallel jobs that runs a simulation replication will need pseudorandom numbers if we're running, say, MCMC. And yeah, then there's the naive way of doing it. It's like, okay, can I just set seeds from 0 to n-1 for each of my workers? And the answer is, of course, no. So if you do that, and this is something you can even try on your laptop, set seed 0, generate 10 random uniform numbers, set seed 1, do the same thing, then 9 of them will actually be exactly the same. So if you assume that your simulation results are uncorrelated, that's not the way to do it. So a better way to do it is to do stream parallel runs, and there's really an entire paper on this, like using simulation studies to evaluate statistical methods where there's much more that I cannot cover here. But basically the idea is to set the seed once, derive uncorrelated random number streams that are constructed in a way that they're 2 to the power of 127 numbers apart in the case of this Lecuyer's random number generator from the parallel package. And basically derive these and then send these to your jobs. So yeah, I still have a minute. This already brings us to the key learnings of this. So yeah, if you really want to efficiently design these trials and iterate on our designs and actually try some different parameters, we don't have a Shiny app to do that actually. So that's definitely something maybe to explore. Then we also need to be able to rapidly iterate on designs. And what I didn't cover here, but is also important, if you have tight timelines for health authority requests on these, you actually want to have that really fast because you have a deadline, a short one. So if you want to try this yourself, profile the code. There's a package called Profis. It's also integrated into RStudio that helps you with that. Optimize the hotspots locally, and then if that's not enough, go to HPC. Can also be HPC in the cloud, and Michael has a demo that we can show afterwards where you can also try, in principle, your workload. Reduce overhead if you find it. And yeah, carefully consider best practices. And we'll also have a course on HPC with R at the Sun conference in September. And yeah, catch Michael and me at the APRO for cloud-based demo. That's all.
Thank you very much, Lukas. Very nice talk. And very nice that I sent an email to the speakers yesterday to please stick to time, and they delivered 3 great talks and all on time. Can I please ask all of them to come, Lukas, sorry, to come on stage for the panel discussion? I will just have 2 questions to start it up and then open. One thing that struck me when I looked at the 3 presentations, that we have 3 quite successful packages, but they were developed in quite different ways. So we heard Diane in an academic setting, Gernot, a company that works with crowdfunding, and then Lukas, the OncoBase is developed primarily by Novartis, I believe. And I just wanted to ask you how you see the potential of these different ways. Maybe we just go in the order. Diane?
Yeah, of course. In an academic setting, you don't have the same maybe resources as in a company, but you have all the freedom. So you can be very creative. We also had the luck to have good advisors, I think, when we wrote the R package. Yeah, I think you have the time. Maybe that's something good. So you have time to iterate. But of course, you also need to have the funding, and so you always have to have the luck before to get a grant. So it has some benefits, but also some downside, I would say. Want to go next, Lukas?
Gernot, maybe we go into order.
Actually, I am very happy that our concept was so successful, because I mean, 6 or 7 years ago, I didn't have this thoughts, because I was very motivated to create an R package, but I also always said, well, but I need money from somewhere and someone. And there was the idea where this working group of the pharmaceutical, how's it called? The working group pharmaceutical research met once, and they said, well, we make something, and there was the birthday of this called service level agreement that we have now made with so many companies.
Lucas.
Yeah, and I guess OncoBase 2, that kind of grew out of a need of having a package that can deal with N drugs, and then M historical data trials, and potentially concurrently running arms, and then also drug-drug interactions, which really wasn't covered by any existing things. So that's how Sebastian then came up with this, with the first versions of the OncoBase 2 package to really address sort of this more immediate need. That, okay, we have this new, more complicated trials, and it's not single drug, it's not a combination, it's like 3+ drugs combined. And at that point, the question was also like, do we continue developing specific solutions, or do we take a step back and solve this in its full generality, basically? And this has been, I'd say, quite successful internally. We have some external users now, but we're also not terribly actively pushing this package, I would say. It's more one that we're also, like, it's on CRAN, it's open source, but yeah, we're not doing very active advertising at the moment for it. That said, it will come to GitHub fairly soon.
Thank you very much. That brings me to the next question. I think one thing that is very nice is that we see increasingly pharma companies publishing stuff on CRAN and on GitHub, and we do the same, and many other companies do the same. Maybe a follow-up question, Lucas, you already started this on. I think of this like you have an invention, it's your package, and then you kind of want that a lot of people use it and trust it, and I just wanted to ask a bit how you achieved that. And maybe, Lucas, you can start.
Sure, I mean, we also have, so first thing is to test it well. So obviously we also have unit tests, but then for the Bayesian hierarchical model, things get a bit more interesting there. It's actually something that's hidden in the package a little bit. In the inst folder, there is a SBC script. SBC stands for simulation-based calibration. And that's actually a way to test whether your Bayesian model fits the correct thing, which is something that actually needs to run on the HPC to really test that. So it's not just a unit test that you fire off on CRAN and hope for the best. It's not going to work. But that's that kind of, yeah, taking testing seriously, especially if stuff like a safety model, that's I would say our first step in that direction, and obviously making it available on CRAN.
Diane?
So for me and for us, it was really crucial to connect to other people. And for the randomization space, we have a really active cross-industry, cross-academia collaboration going on with people from Novartis and from Boehringer as well. Where we meet, I think, once a month and really address questions like where else can we advertise this package? So now we have user groups for example, for ASA. We have a website, and we write papers. We really try to also do white papers that might be adopted by regulators. And I think for randomization, it's really important to get the trust from the regulators, because I heard earlier when regulators tell you, no, you can't use these other randomization procedures, then your company will never adapt it, right? And so convincing the regulators is really key.
Thank you, very cool. Gernot?
Oh, I've shown the CRAN download stats from our package. It was 1,200 per month around. And that's obviously some kind of limit. Pharmaceutical applications or statisticians are using such packages. We want to have our package also applicable to other people. For example, as I said, psychologists, and they are using at the moment obviously other, they're doing a lot of sample size calculations. How you say? I say psychologists. They are also performing sample size calculations, but they are using programs which have a 10 times more download stats from CRAN. And actually, I do not understand. And I try to make three things: advertise, advertise, advertise. Our package to make our stats even bigger.
Thank you. I have a couple more questions in my pocket, but I'd rather hear what the audience. Hans-Jürgen?
what you have done is really amazing. I mean, the cornerstone of clinical trial is randomization, but if you think what we as pharma companies have done during the last 30 years, to a certain extent it's a scandal. There is nothing, typically there's nothing available. The far majority of trials, even let's say confirmatory clinical trials, are done with blocked randomization, with fixed block design. And the typical situation, is that the statistician doesn't even know what's going on, because all this is handled as part of the packaging part of the company. So I'm so glad, and it's so good what you have done, and I'm also so glad that you have this cross-industry working group, which really a nice paper, nice white paper has been published. And I can also promote this, and let's say from the other companies, look at this package, look at the white paper. This is really worth adopting, because there's also one other point. It's so easy to connect this randomization app to the available packaging software. So connecting interface is not an issue at all. So to a certain extent, there is no excuse to using innovative and efficient randomization methods.
Thank you.
That was a very nice plug. Thank you. Any other questions from the audience?
All great presentations. I'm wondering if you could maybe talk to how, I mean, you've kind of, I guess you all individually kind of were the genesis of the idea to get are involved in the start of the project, and I'm wondering how you managed to get more people involved and contributors involved, not just users, and build, so it's not just you sort of pushing this forward all the time, and how do you get more people to kind of join the party?
Maybe we can just start. So for me, it was really to connect to other people who were active in the space, to be really proactive and say, oh, I see you have a really good publication on this topic. And then also just put myself out there a lot, and then people actually came to me and asked, can we work together? Being proactive.
So,
yeah, I guess I can build on top of that. Also, I mean, we gave a lot of internal trainings, for example, for Oncobase to drive adoption and also get feedback. That was, let's say, internally fairly popular. We haven't really done this externally so much yet. Sebastian has started this with RBest, which you might also be familiar with, which is sort of from the same corner of the company. So yeah, giving trainings, I think, is also a good approach.
Definitely, yes. For me, I mean, I'm working on adaptive design since 30 years now, and my focus was always on software. And I mean, I have also at that time created AdPlan, but I saw my experience was that licensing is a difficult task, and I didn't want to do it anymore. And so free software, freely downloadable and really for free is exactly what is nowadays appropriate.
Thank you.
I just have one question for you, if I may. So you mentioned several times the service level agreement. Could you reveal maybe some core components of such agreement? What is about availability of the package or something else?
The package is for free. Everyone can download it.
Yeah, okay. But what is inside the service level agreement then?
The service level agreement, especially one essential point is the free training per year. Okay. And it's also that you are in this RPACT user group. We have a yearly meeting where we come together and discuss possible enhancements of the software and give some a yearly report of our work, for example, such things.
Thank you very much.
Maybe I can add to that. I mean, if you think about we in industry, the problem for us with academic packages is the huge validation, to be documented validation effort, which is not there. Even if it's programmed in a perfect manner and everything is there, we need to convince our own quality people, our own auditors. Therefore, let's say if we want to use such a sophisticated statistical package for confirmatory trials, and these confirmatory trials cost hundreds of millions of dollars, yeah? So we need to be 100% sure and documented that what we are doing is really correct. And this, of course, puts a huge effort on the programmer, which typically you as an academic, if you develop a package, you don't get rewarded for that. And that's really the point where it makes sense to say, well, if you're an academic researcher, you get it for free, RPACT. But if you're a pharmaceutical company and you're making $1 million business with that tool, I mean, it's only fair to enter an SLA, and then you get the full documentation, and if the FDA comes, and if your auditor comes, the inspector comes, you have it. And they will even support you if they have questions. So this is really, I believe this is really good value for a pharmaceutical company.
Can I just chime in there? We had earlier the discussion about the money, right? And that the money is not the motivation. But I think there is some motivation in getting the money to be able to do what you love and what you're really good at. And I think this is a prime example for that. Yeah.
Other questions? If not, I have one. So we talked here about design of clinical trials, and there's quite a lot on CRAN. We've talked already, I don't know, 20,000 packages. There's a task view for clinical trials, but that one is also maybe not the end of all. For example, OncoBase 2 is not there as of yesterday. So I was wondering, do you see another resource where these really high quality packages are kind of properly advertised? And B, do you see any gaps in the kind of design landscape, something that the world is waiting for still? Gernot?
I actually saw a gap, but there isn't. I saw a gap in blinded sample size estimation tools, but I actually now know 2 already that are available on CRAN. Are you available on CRAN?
We are not yet on CRAN.
But not yet on CRAN, but GitHub or?
I would say I have seen others as well for blinded sample size.
Yeah.
We cannot compare with the ones that are already on CRAN.
Okay,
so we are a small contribution compared to those that are available, and just maybe the visualization comes in additionally.
So that's not a real gap, but maybe a small gap. So it should be maybe completed.
Okay. Lukas or Diane?
I guess not in terms of gaps, but like thinking about where we would find high quality packages, that's maybe something we should collectively try to address. Yes. Like we heard about risk metric today, and yeah, what kind of packages would we be comfortable using for certain use cases that have, like there might be no risk at all, like if I want to analyze some data from my solar system, who cares? But if there's patient lives at stake, then the effort should probably be a bit different.
So for me, I try to always look at high quality documentation. Mm-hmm. And one resource I use frequently is the Journal for Statistical Software. So if a package is documented there, I know that it's good quality. but it also takes a long time, or at least when I tried to publish my paper, it took like 2 years to get in there, and so it's kind of outdated, I imagine, but probably there are much more packages out there that are already well established, but just didn't make it through the publication phase. And in terms of a gap, I think there are now more different types of designs, like umbrella and platform, and like in general master protocols. I know there are packages out there for that, but I think in terms of design, they're still basic. They're probably all using permuted block randomization. So maybe there is something there that can still be done.
Thank you, very good. I mean, just to bring up what Gernot said about sample size calculation, I mean, RPACT is great. As you know, I'm a big fan, but it covers only binary, continuous, and survival.
You want to have count data, I know.
And so I think just for a basic all-purpose sample size calculation software,
yes,
there still need maybe something for RPACT.
we discuss this in September.
Yeah. Cool. James, wait for the mic, please.
Sorry, just extending that question which Marcel asked. So on the clinical programming or statistical programming side, you have the Pharmaverse now, which is like a grand task view plus a community plus leaders getting together to come up with strategies around what's the gaps. I guess I'm kind of interested what your thoughts are on. Statistics has been doing our packages for a really long time, but is there a place for something similar, or even talking to the Pharmaverse about extending? Like, I guess I'm asking, could there be a biostats verse, or even re-scoping the Pharmaverse if they agree?
Who wants to take this one?
Yeah, I think that's an excellent question. I think there is definitely an opportunity to talk to the Pharmaverse people to extend the scope. And I think packages or verses like that are a great opportunity to integrate the methods further. I think that's then the challenge, right? That you need to integrate the methods that you've been using, and maybe they don't use the lower camel case. Maybe then we have to kind of modify our procedures, but that's probably not a high toll to ask compared to the visibility and standardization that we would get from that.
What I found very interesting from our concept of crowdfunding was that now pharma is working together. So, I mean, you see in Basel very well that there are pharma companies, but also in the working group for pharmaceutical research, there's also this working group for different pharmaceutical companies, and they are working together, and we are working with them together. Right? I think that's a good point to further develop such packages. Yeah.
I couldn't agree more. I mean, since I joined Pharma again 7 years ago, that has been one of the pleasures of my work, how good the collaboration is, that I'm not only working with Roche colleagues, but also Novartis, BI, many other places, and academics. So I think it's become much more open. Yes, Ursula?
I think a gap that I would see, and actually even within Cytel we are discussing that, what is difficult at the moment is sometimes I use GS Design, RPACT, East, and so forth, different design instruments, but when I want to compare the simulation results from across the different platforms, I cannot. And even if we would have a C-disk for simulation results to some extent at least, we would be able to do visualization tools that collect the results from various different designs and then compare them all together. And I think such a standardization effort would be really good, because one of the problems of the blinded sample size re-estimation in East also is it cannot be simulated. You cannot compare whether you do better with an unblinded sample size re-estimation or a blinded sample size re-estimation, but the operational process that is behind it would be much different. So the costs to the clinical trials would be much different whether you do it blinded or unblinded. So this across-platform standardization of output would be the easiest way to make different tools available, but still have the opportunity to really compare the results one-footed.
Thank you. I totally
That's
agree.
exactly what we will have available in our Shiny app. There's a compare designs button where you compare the different designs you have selected.
Within RPACT or also within GTS design?
That's within RPACT only. You want to have between the packages, okay. Mm.
But within RPACT is already good. I mean, better than what we have in EASY.
So we have maybe time for one last question, and please wait for the microphone. So if there's nothing burning, I think I would like to very much thank the speakers again for a very interesting session.